So in this video, we're going to focus on membrane potentials. Like all cells of your body, neurons have a resting membrane potential, also called resting voltage. And what this means is basically a separation of charges across the plasma membrane. So what we find is that there's going to be typically more negative charge on the inside of the cell than outside of the cell. And this is due to ions, whether they're ions like chloride or sodium or potassium, or even, charges that you find on things like protein. But what's weird about neurons is that unlike most other cells, neurons can rapidly change their resting membrane potential and therefore rapidly change their voltage. And this is done in an excitable way. So neurons will change their voltages in response to a stimulus. Whether that's another chemical or electrical stimulus, we say that neurons are highly excitable. So what we find is that opposite charges across the plasma membrane are attracted to each other. And there's energy that's required to keep these opposite charges separated across the plasma membrane. This is why we call this the potential, because there's a potential for these charges to move. In fact, energy is liberated when these charges move towards one another. Now, when opposite charges are separated, the system has potential energy, and this is essentially what we call voltage. So voltage is a measure of potential energy that's generated by the separation of charge, and we can measure this in volts or millivolts. So with respect to the human body and our cells, we're going to measure this on the order of millivolts, which is, basically very, very small separations of charge. Now this potential difference or potential is ultimately what is the resting membrane voltage or resting membrane potential. Now the greater the charge difference between the points, the higher the voltage. So when we say that there's a big difference in charge, like maybe you have a tremendous amount of negative charge inside the cell and, some more positive charge outside. If there's a large separation, then you have a higher voltage, just like a larger potential difference. So current is the flow of electrical charge, and in this case, ions between 2 points. So the current that flows through our cells isn't the same as like electrical current through a wire, because that's the flow of electrons. But rather, when you have ions that are flowing through channels across the plasma membrane, that's what we call current. And this can be used for work. So flow is dependent on both voltage and resistance is basically the hindrance to charge flow. Now, insulators are substances with high electrical resistance. And so an example of an insulator would be, the myelin sheath around your axons. It functions as a good insulator because it has high electrical resistance and therefore doesn't allow for flow across that membrane. And therefore, those ions inside the axons can only flow in, a very particular direction. Conductors are substances with very low electrical resistances. So, for instance, the inside of the axon is a really good conductor, but the outside layer, sheath around the axon, myelin, is a really good insulator. And that way, we actually can conduct that current or flow through the axon rather than outside of the axon where the information might get dissipated or lost. So Ohm's law gives us the relationship between voltage, current, and resistance. So we say that current or flow is equal to voltage or potential difference divided by its resistance. So we say that resistance and current are inversely proportional, because if you increase resistance, you decrease current. If you decrease resistance, you increase current. And voltage is that potential difference or separation of charge. This law comes up a lot in physiology. When we talk about blood flow and airflow, it turns out that those equations for flow of air and blood are basically Ohm's law. Now, current is directly proportional to voltage, so that we find that the greater the voltage, the greater the current. And if there's no net current flow between 2 points with the same potential, then we say that that voltage is zero, so that there is actually no current. So you've got to have a potential difference in order for current to exist, and these are proportional. However, current is also inversely proportional to resistance. So it means they vary in opposite directions. So if you increase resistance, you decrease current. Now, the role of membrane ion channels in the plasma membrane of your neurons is they basically help to establish this resting membrane potential. Now, there's large selective proteins that serve as selective membrane ion channels, and at rest, potassium channel are mostly leaky, and so that potassium flows the most while these neurons are resting. And ultimately, there are two major types of ion channels. We got leakage gate channels and gated channels. Leakage channels are always open, which means they always allow for some current across the membrane. And gated channels are conditionally open. These might be chemically gated, voltage gated, or even mechanically gated, which means that due to a change in a condition or state, like a chemical or a change in voltage or even mechanical stimuli, can open up these channels. Now, chemically gated channels are things like ligand gated channels, which open only when a specific molecule, like a neurotransmitter, binds to this receptor channel. Voltage gated channels open and close in response to changes in voltage or membrane potential. And mechanically gated channels open and close in response to physical deformation. You find a lot of sensory receptors are actually mechanically gated. So things like pressure and vibration are mechanically gated channels. So chemically gated channels only open in response to a chemical. So you might have a neurotransmitter bind to this receptor here, and this can open up a channel that allows for sodium to flow through. We saw an example of this back at the neuromuscular junction where the nicotinic channels or acetylcholine channels were like this. So you needed acetylcholine to bind to this receptor, allowed the channel to open up, and allowed it for ions to flow as a result. Voltage-gated channels open and close in response to changes in voltage. So at rest, you might find that there's more negative charge inside the cell versus outside the cell, and that these channels can actually open up if this switches, where if you have more positive charge inside the cell, that might cause these channels to open up and allow for ions to flow. So when these gated channels are open, ions diffuse pretty quickly. In fact, these ions will diffuse along their electrochemical gradient. There's a chemical concentration gradient for these ions. So you might find that sodium is in higher concentration outside the cell and lower concentration inside the cell. And there's also an electrical gradient as well, where you have a separation of charges across the membrane. So that also plays a role here with diffusion direction. So the electrochemical gradient is basically electrical and chemical gradients combined. Ion flow creates current, and voltage changes across the membrane as a result. Remember, this actually can be expressed by rearranging Ohm's law, where we can say that voltage is equal to current times resistance. So as long as you have a current and there's a resistance present, then you have a voltage. If there is no current, then voltage will be zero. So what we find then is that voltage represents the movement of ions and the resistance of the flow of those ions. And all of these are actually proportional. So we find that voltage is proportional to current. Voltage is also proportional to resistance in this example. So a voltmeter can actually be used to measure this potential difference. So scientists can actually measure the voltage or separation of charge across the plasma membranes of neurons and other cells of your body. Resting membrane potential, or RMP, is basically the voltage inside of your neurons while they are not currently generating an action potential. And we find that resting voltage can vary from like -40 to -90 millivolts, but it's usually approximately around -70 millivolts or so. Because of a separation of charge, we say that the membrane is polarized. Now, this potential difference in charge is actually generated by a variety of factors. For one, it's generated by differences in the ionic composition of your ICF and ECF. You find that in the intracellular fluid, there's different compositions of ions. Like you find more potassium and more protein inside your intracellular fluid. And in the extracellular fluid, you find a lot more sodium, but far less protein. And we also find that due to differences in plasma membrane permeability, only certain ions can actually flow at any moment in time. And due to the flow of those ions, that generates a current, which changes the voltage. So we can use this voltmeter and basically measure the difference between the inside and the outside of the cell. Now, what this voltmeter is telling us here is that relatively, the inside of our axon here is more negative with respect to the extracellular fluid. And this potential difference or separation of charge is due to, basically a difference in the composition of the extracellular fluid versus the intracellular fluid. But remember, neurons can change their voltages, so that if this were like a red blood cell, you might stay at -70 millivolts for forever as long as that cell is living. But even because neurons can change their voltage, this value can change as a result of ions flowing across the membrane. But what changes the flow of ions are basically a change in the permeability or the opening and closing of certain membrane channels. So the differences in ionic composition are laid out here. We find that the extracellular fluid has a higher concentration of sodium than the intracellular fluid, and this is balanced by chloride ions. Intracellular fluid has a higher concentration of potassium than the extracellular fluid. This is balanced by negatively charged protein. Now, potassium plays the most important role in resting membrane potential because it's the leakiest while at rest. So what we find in here then is that this concentration difference is actually established by the sodium-potassium pump. So remember, the sodium-potassium ATPase uses 1 ATP to pump 3 sodiums out and 2 potassiums in. Now, if you let this run over time, you find that eventually the extracellular fluid is going to have a significant amount of sodium, and the intracellular fluid is going to have a significant amount of potassium. Now, the concentrations are pretty well balanced. You find about 140 millimoles of sodium in the extracellular fluid and about 140 millimoles of potassium inside. Now, but they do have a gradient though. So you see that extracellularly there's only 5 millimoles of potassium, and intracellularly we only have 15 millimoles of sodium. So that if you allow these ions to flow down their concentration gradients, potassium would want to flow out and sodium would want to flow in. However, this is the sodium-potassium pump, which isn't allowing these ions to flow down their concentration gradient. Rather, this is a primary active transporter, which uses ATP to pump these ions against their gradients. And that's effectively what keeps sodium low inside of the intracellular fluid is the fact that it's being pumped out. And what keeps potassium low in the extracellular fluid is the fact that it's being pumped in. So differences in plasma membrane permeability can occur with opening and closing of different channels. You find that at rest, sodium is slightly leaky through leakage channels. However, potassium is 25 times more permeable than sodium at rest. So as a result, potassium diffuses out of the cell down its concentration gradient, and because you're removing positively charged potassium from the inside of the cell, it makes the inside of the cell relatively more negative. Now, more potassium diffuses out than sodium diffuses in, and as a result, the inside of the cell becomes more negative with respect to the outside. Now, this is what the resting membrane potential is basically generated by, and this ability for these ions to flow is actually maintained by your sodium-potassium pumps, which maintain these concentration gradients even though that leakage channels exist. So remember, the sodium-potassium pump uses ATP to pump 3 sodiums out and 2 potassiums in. So although potassium is diffusing out of the cell, it's also being pumped back in by your sodium-potassium ATPases. So just to kind of summarize what this looks like is we have leakage channels as well as your sodium-potassium ATPase. Now, if you can isolate the effects of voltage due to the flow of potassium, we'd see that potassium, because you're removing positive charge from the cell and potassium is flowing down its concentration gradient, the inside of the cell becomes relatively more negative. In fact, if only potassium flowed, it would get to about negative 90 millivolts. Now, if you throw sodium in here too and have a sodium leakage channel, which remember is only 1/25th as permeable as potassium, you're bringing positive charge into the cell as well, which makes the cell slightly more positive than if it were just potassium flowing. So we find that as a result, then our resting membrane potential is actually negative 70 millivolts. And it's basically a balanced voltage in response to potassium flowing out and sodium flowing in, that this is the voltage we get inside the cell. Now, this concentration gradient is maintained by the sodium-potassium ATPase over time. So resting membrane potential is basically due to the concentration of ions across the membrane and the permeability of those ions. So the membrane potential can change with graded potentials and action potentials. Remember, graded potentials are incoming signals that operate over short distances. These things can degrade over time and distance, so they get smaller. Action potentials are long-distance signals that actually don't get smaller. They stay the same size. We'll make sense of this later. Now, changes in membrane permeability are used to basically transmit information. So that when we say that electrical information is transmitted along the neuron, ultimately this relates to changing the membrane potential, and this change is a piece of information that our neurons use to communicate. So just some different terms to describe voltage changes. We have depolarization and hyperpolarization. Depolarization is when the voltage of your cell becomes slightly less negative than resting membrane potential. And hyperpolarization is when the inside of your cell, like the voltage of your cell, gets more negative than resting membrane potential. And so if you think about these changes, ultimately if your neurons are depolarizing, this increases the probability of causing an action potential. if your neurons are hyperpolarized, this actually decreases the probability of producing an action potential. So what we find here then is basically, if you have a depolarizing stimulus, voltage becomes more positive, but then it can actually go back down to rest. And if you have a hyperpolarizing stimulus, voltage can become more negative, but then it can go back towards rest. Remember, rest is this voltage that you find when neurons are not conducting any kind of currents, deliberately rather, and this is the resting current that's due to their sodium leakage channels and your potassium leakage channels taken together makes this negative 70 millivolts.